So here are some results that were generated within the GEroNIMO project, but also previously in the GeneStretch project. So here's some of the work done by Bruno Perez, and this is in this 2022 paper where we first introduced this, where we call the GTC blob, what shall call GMC blob here today. So what we had here is we had a data set on mouse, and we had phenotypes, different phenotypes available for these mouse. But I'm here just presenting some results on predicting body weight at different ages. And what's important is we had SNP genotypes on these individuals, but we also had gene expression, which was measured in the liver, and that was measured at week 26 of age. These mouse, they're called a diversity outbred population because some 20 generations ago, people started with 8 almost fully inbred strain of mouse. And then they just started to cross between them, and they continue to do that for many generations. But even today, they're still continuing this. What this setup allows to do is that in all the later generations, we can track all the different chromosome segments of these mice back to the inbred ancestors. So if you genotype them at a medium density panel, you can actually just infer the founder haplotypes and impute that completely.
So we have got a question about this exercise, the exercise before. The accuracy we obtain with methylation learning is perhaps higher than what I obtain with genetic values.
That may be, yes.
Why when we look at the genetic variance, the genetic variance the variance explained by methylation related to the total is the same magnitude order of the one of genetic values, and why the accuracy is almost twice higher than the.
Let me finish this. I'll get there.
So sorry, because
no, that's fine. But
because moving into my mind. So
yes, yeah. So what we did here is actually the exercise that you've also done today is that we constructed a number of different models. And we first looked at the variance partitioning, right? So here we predicted body weight at 3 ages, 10, 15, and 20 weeks of age. And we either did, in blue, we had a G-blob kind of model. So there we see that the SNPs explain something like, on average, up to 40% of the phenotypic variance, right? If we then, in the middle, we use the gene expression, so we call this T-blob for transcriptomic blob. Actually, the proportion of variance explained with the gene expression was quite a bit higher than with the SNP genotypes. So that's one thing to observe. The other thing is that to the far right, we have both in the model, and there we had this conditioning approach. So you nicely see that indeed we were able to have this G component explain as much variance as if it were just in the model all by itself, right? And then the T component or the gene expression was able to recover phenotypic variance that was explained by the T blob, but not with the combined one. And what is the other observation here that we can make is that if we use gene expression that was measured on 26 weeks of age, so when the phenotype was measured at an age close to the moment of measuring the gene expression, it also explained more variance, right? So biologically, this seems to make sense. But it was also nice to see this. So we did this, the variance partitioning, but then we also did genomic prediction, actually in a rather similar fashion as you've done this afternoon with a cross. Oh no, here in this case it was actually a forward in time, so it was the youngest generation predicted using the older ones. And then we did the same models, here. So there's bars for each of the different models next to each other. And what you can see is that the blue bar is the lowest one. So with SNP genotypes alone, we have the lowest accuracy of predicting phenotypes in this case, right? Then with yellow, if we only use the gene expression, we do considerably better. And if we combine the two, we actually don't do any better than using the gene expression alone. So in this case, it seems if you're interested in predicting the phenotype, you don't need the genotype, but the gene expression is what gives you most information. Yes.
If you were to select those mice, some trait, based on what would you select them? On the G component?
If you're interested to do, let's say, genetic selection and make the next generation better, I would select on the G component, yes.
Would it be the G from which one? From the GT block?
I think that the two should be quite similar because in this case, the T was conditioned on the G. So I think that the breeding values from both models should be quite similar. So it shouldn't really matter. Yes. Yes.
Expression is species. So for each trait, you have to choose the right tissue or what was used for the body weight.
Here it was liver. What was measured in the liver. The data was available, it was generated, and we just made use as how it was available. But we can understand that maybe the liver has some functioning and growth, right, to some extent. Yes.
Would you expect lower variance explained if you sampled the brain or the ear versus?
Probably, yes. Yeah. Well, this is, I think on this mouse population, they have maybe phenotypes of 80 different traits, like they look at cholesterol in the blood, they look at the weight of heart, they at bone mineral density, like you can't think of what they're all looking at. Just study this as sort of a model organism, right? So we developed these models, and at that time we didn't have the data with all these different components, so this was available and we used it. But it worked out nicely in the end. So here, in terms of prediction accuracy, so it's higher for expression than for genotypes. And again, for the later phenotypes, so that were measured closer to the moment where the gene expression was measured, the prediction accuracy for the phenotypes was higher. So that seemed to make sense also. But there was no increase when we combined two source information. So that's one set of results. Another one is work from Levi here. What we have in the GEroNIMO data is this was data from Danish pigs. It was a little bit short of 450 pigs from 3 different breeds, and there was phenotypes of 4 different traits: average daily gain, residual feed intake, back fat thickness, and strength. And we had pedigrees available for them, 50,000 SNPs. And from the muscle tissue, DNA methylation measured using RBS, but also RNA-seq data was available. So that's quite a neat data set, because the data set is not that big, but to have both this RBS and the RNA-seq made it quite interesting. So what Levi has done is he built all these different models, and so this is for the trait residual feed intake. And in first instance, he included all the CPGs, the methylation scores of all the CPGs with some minimum filtering in the model. And then there was a B blood model, so pedigree blood, G blood, T blood, so that includes the transcriptome or the RNA-seq data. M blood included the methylation, and then it was all different combinations of these 2 or even the 3 three layers of information that we have. So the values in here, they are accuracies presented as percentage. And what you can see here is something which nicely aligns with the results that we found in the mouse data, is that if we compare the accuracy of G blood and T blood, is that the gene expression just explains the phenotypes much better, right? So this is, the top is in all breeds, across all breeds, ignoring the different breeds. Not completely ignoring, but combining them all together in the analysis. And at the bottom, it's only the GEroNIMO, so then we have very little data. But if we did this, here we have 44% accuracy with T blood and only 14 with G blood. So we see a very similar picture as with the mouse data. Yes.
Yeah, for G block, we are using all the SNPs.
All the 50,000. Yes. Well, actually, I think in the top one, you only used the ones that were segregating in all three breeds, and it was a panel of.
So it got a bit less, only 10,000.
Right.
The SNP chips were 50K, but because they were different SNP chips and because of differences in breed, then the intersection of the SNP was 10,000.
Are you trying to use only the markers that are associated with the trait?
No. Well, the numbers were already small to start from, right? So if you would do that, yeah.
Because in the T lab, you are using the expression of the genes that are differential expression.
No, all the genes,
All the genes.
right?
Okay.
Yes.
14,000.
Yeah. So for the methylation data, at first instance, the 1 million CpGs were used. And what you can see here is that the M blood across the breeds, it did a bit poorer than G blood. And it performed within the breed, it performed very badly. We actually got a negative accuracy. So it didn't seem to be very useful at all. So then we realized, yes.
Question? Sorry.
Yeah.
So earlier you mentioned the diminishing returns when we add more and more SNPs to our data set and in terms of the G blood, but have you looked at that within the methylation data? So is that all the same trend?
We didn't do that exact same exercise, but when we looked at these results at some stage, what we realized is that, as you've also seen, if you look at the distribution of the mean methylation score, it's very skewed, right? There's very many are close to 0 or 1, and there's very little variation in a lot of them. So we then filtered for that all the CpGs had to have a certain amount, certain variance with them. And that reduced it down to 16,000. And if we did that actually for the all breed analysis, it didn't really affect the envelope results, but then at least within the breeds, then we got a proper positive accuracy that seemed to make sense, right? So here, well, if we do that, we have maybe a little bit over 100 individuals to estimate effects at 1 million CpGs, and that's also a bit stretching what is possible. But so here we see that doing this filtering based on variance was quite relevant.
Do you do the filtering based on the variance of the 3 breeds or the variance of does that make sense?
Was the top, the ones that had the most variance in each breed, right?
Okay,
And then I take the intersection.
Right.
50,000 most variable CpGs in each breed. Then I take the intersection of these 3 sets, and then it comes down to 16,000. Because it's different. The most variable CpGs in one breed are not the same.
Yeah. So you want to make sure the CpGs you included vary in all 3 of the breeds.
Correct.
Yes.
And in the respect that we have also kind of interaction between the T and M. So the GTM is a good interaction or no interaction?
No interaction.
Okay.
No, it's just the GTM is just like we have the GM block this afternoon, but then there's also the T component added to that.
Yeah.
And the same for the GT blood, just the two combined GM blood, like we did this afternoon. And G by N, that's the like interaction that we also did today.
Yeah, so T is for transcript, so it's RNA-seq.
Yeah, and methylation are linked because it can repress the expression of genes with methylation. So I guess biologically, at least, they are probably linked. So considering which part is due to which, I probably have some confounding methods.
Yeah, so I think that's a fair point. So from the onset, when we started the GEroNIMO project and we start develop these models, indeed, part of our reasoning was methylation can regulates gene expression, right, to some extent. So in that sense, it also makes sense that if you have the gene expression rather than the methylation, that you have more information, right? Because you're closer to the phenotype in a way.
So I think that would behave better than gene typing data, but this was performed with SNP chip, right?
Yeah,
Okay.
yeah,
Maybe the SNP chip isn't well adapted for this breed. Okay, it's Duroc, but what were the other breeds?
so the other breed were Landers and Yorkshire.
So they should be.
Yeah, so indeed, as you probably know, Landers and Yorkshire are genetically similar. But I don't think Mario has done other studies with T blood, and it consistently does better than the SNP. So seems to be gathering evidence that it's better for prediction than these.
So in the paper by Valentin Haas that I mentioned earlier, he had data on quails. He didn't have methylation data, but he had genotypes and gene expression, and also in his case, the gene expression explained more than the SNPs did. Yeah, but again, this is the accuracy of predicting phenotypes, right? So I'm not claiming that with gene expression you can get better predicting. That's the.
And this is probably still very much tissue dependent now.
Sorry.
This is probably still very tissue dependent.
Yeah, so I guess in most cases here where we measured the gene expression was in a tissue that was relevant for the phenotype. Yes.
So trying to apply this to an actual breeding program, the G blood still is probably more universal and
Yes.
predictor versus.
Yeah, because also I think I said at the beginning of today, if you're going to use these other omics, you know, which tissue are you going to measure them, which moment of the day, etc. Right? That's going to be quite difficult. And also it may be difficult if it's invasive, right? For the mouse, it was measured in the liver. Now, I don't think you want to take functions of livers in routine breeding programs. But, so we worked on this data here in Wageningen, or Levi did, and a group in ITRA that's also involved in GEroNIMO. They applied a number of deep learning, but also feature selection models on this same data. And well, there's a lot of results here, but the main thing here is that, or the main takeaway is that if they used partial squares to prioritize or to select which features to use, so which CPGs, which SNPs, which genes the expression should be included in the model, then that was where they could achieve the highest accuracy compared to any other model that tried to include, for instance, all the features or all next to each other. So, but yeah, these results are fairly recent, so we now try to compare how does that do relative to the mixed model predictions that we have generated. So just to wrap this up, it seems to be important if you want to use, for instance, methylation for prediction to do some sort of feature selection. And in our case, we saw that just taking the more variable CPG sites is an important step to make. And not only for prediction accuracy, but also to get more robust results. So what I showed is that within breed predictions, we saw negative accuracies, which, well, basically mean they're completely meaningless. But then selecting for variance turned this into something meaningful. And also, if you combine different breeds or you go across breeds, it seems that there using multi-omics information is also can be quite useful.
This has been proven. That's across.
This is just based on the results that I've shown that like, this seems to be more beneficial if we combine those different breeds. But this needs more proof by other groups and other data. Yes.
Because of linkage equilibrium, which is specific to each breed on genomic data.